tired, and you feel pain because you're yawning or something like that. So that's not the kind of pain we're going to talk about. What we're going to talk about is damage-induced pain. So we're talking about somatic pain here. The receptors we're talking about are free nerve endings. So free nerve endings are going to detect pain. There are distinct receptor types that are trip channels, and some of the different types of stimuli would be extreme mechanical, like ripping your axon or cutting it, extreme heat or cold, nauseous chemicals, polymodal nociception. If I had a band, I would call it polymodal nociception. I think that would be so cool. That's mixed. So that's mixed detection of pain. So in every case, you have activation of receptors. Maybe you're just ripping some channels open. You're going to have some receptor potential. In this case, it is depolarization. And then action potentials in the primary afferent fiber that's going to carry that information to the central nervous system. So we're going to consider the extreme mechanical in more detail. So the mechanical detectors are, again, trip channels. So you're seeing trip channels a lot. So we have a couple different classes of channels. We've had voltage-gated ion channels. We will have ligand-gated ion channels. The trip channels are transient receptor potential channels. So again, they're ion channels. And when you physically damage these, they're going to start to change your receptor potential and start to send a signal. They're also going to release proteases and other substances into the area. And these are going to be very important themselves. So the actual, you destroy a cell and you release a bunch of proteases, and proteases cleave proteins. So the proteases are going to cleave proteins into peptides. Peptides are a class of neurotransmitter. As we'll see, there's a lot of different peptide neurotransmitters. And one of these is bradykinin. So bradykinin is a very, very potent pain-inducing substance. So you've destroyed a cell. You've released proteases. It cleaves pro-bradykinin, so not processed bradykinin, into bradykinin. And then bradykinin can elicit pain. There are bradykinin receptors throughout. We will mostly focus on B2 and B1. So bradykinin receptor 2 and 1. 2 is expressed constitutively. So when you say something is expressed constitutively or is located constitutively, it just means it's there. So you would find these throughout the body. They have a role in vasodilation and pain stimulation. I just put this in here. So ACE inhibitors, so angiotensin converting enzyme inhibitors, actually cause bradykinin to build up. And that will cause some of the side effects that class of drugs can have. Now, B1, the bradykinin receptor 1, will be expressed in damaged tissue, and it can play a role in chronic pain. So chronic pain is a terrible problem that people can develop, and they will experience pain in response to stimuli that shouldn't be painful because they're upregulating these B1 receptors. So B2 receptors throughout the body, and then if you have damage, you can upregulate those B1 receptors. Why would you want to do that? What a pain. Why have these receptors if they can cause you chronic pain?
Stop you from damaging it more.
Yeah, good. So if you have damaged tissue, you probably want to make sure that tissue has an opportunity to heal. And so if you upregulate some pain receptors, you'll be more sensitive in that region and hopefully less likely to damage it. I always thought this was sort of surprising that a lot of really nasty diseases, Hansen's disease, which is what we call leprosy, is really because you just don't detect pain anymore in your periphery. And because you don't detect pain, you actually will get little cuts and whatnot, and you walk on them, and they will fester, and you'll lose like limbs and stuff. So it's really just because you're not detecting that is going to be induced. So very important here. In any case, you have these receptors, and if you stimulate those receptors enough, you'll trigger an action potential, and the action potential is going to go to your central nervous system, and maybe you'll have a reflex, and then it'll reach your brain, and you'll say, "Ow, that hurt." So that's part of it. That's part of how we would detect pain. But then there's also another component. And this is really unique to the primary afferent fibers that are detecting these stimuli. It's something called an axon reflex. So Fritz had a question about the organization of the nervous system, and I said, well, we have this generic idea that you have dendrites, and they detect signals. He actually said this, and then the cell body is connected to the axon hillock, and that's where you trigger the action potential, and it goes down the axon. But in reality, there's a lot of variations. And one example would be these your naked nerve fibers that are detecting pain. So their actual receptors are on axons. So they're this sort of funny type of neuron called a bipolar cell, and they have basically two long axons. So if you have injury, one year I tried to project on this board, and so if this wasn't there, that would be awesome. I could just, if they would just make a board that was like a foot higher. So imagine you have your free nerve ending, you've got the cell body, and it's going to go to the spinal cord and brain eventually. And you've damaged this part of the axon. And what results from that damage? Maybe you've damaged this cell right here. Let's leave the axon intact. You've killed this cell, this poor cell. You've ruptured it. So proteases are going to be released, and they're going to cleave, eventually cleave a protein into bradykinin, and that's going to bind the receptor. And that was a trip channel, so that's going to change ion conductance. It's going to cause some depolarization. You can eventually have enough depolarization that you're going to fire an action potential, but you also have this axon reflex. And the axon reflex is actually the signal traveling to the end of the axon, so it invades the adjacent terminals of the axon, and it causes release of something called substance P. So the axon reflex involves release of substance P. In addition to polymodal nociception, I think substance P would be an awesome name for a band. I always do a Google image search and just hope that I'll find some former WWAMI student who's dropped out of the medical profession and started a rock band. So you have this axon reflex, and the axon reflex is going to involve release of substance P from all of those axon terminals. Substance P, in turn, is going to bind receptors on mast cells. So we've switched systems. So mast cells are cells of the immune response, and they're going to release histamine. So histamine is going to act back on the original pain receptors and make the pain worse, so it's an amplification system. And it's also going to act on nearby capillaries and cause dilation. So this is why we would get the swelling and redness associated with injury. And this partly accounts for the different phases of pain that you would feel, like if you had fast pain and slow pain. But most of this actually turns out to be the fibers that actually carry the signals to the central nervous system. So if you had fast versus slow pain fibers, if we were to break them up, what do you think the difference is? So fast, what would a fast fiber be like? Myelin and bigger diameter. Good. And so your different fibers that carry information into the spinal cord are A alpha, A beta, A delta, and C type fibers. Now, A alpha and A beta, we're going to put away a little bit. This is more proprioception. The A delta fibers are going to carry fast pain. The C fibers will carry slow pain. I bet that's the only time you'll see that. But the principle itself is important. If you were to block these, so red represents myelin here, so this is our fast fiber. If you were to block this and plot out just pain, if you were to chart out pain in someone, you'd have this fast component of pain, like, ow, I hit my finger, and then later you'd have that more dull, long-lasting pain. If you were to block the myelinated fiber, you could block that rapid phase, and if you were to block the unmyelinated thin fiber, the C-type fibers, you would block that second wave of pain. So again, just to show what this would look like in comparison, you have different fiber types. So we name these A alpha, beta, delta, and C-type fibers. And this is just an example of some of the things that they would carry to the central nervous system. So proprioception here, and then your three nerve endings, so your pain receptors here. And then if you were to look at their diameter and their degree of myelination, you would see that the slow fibers are indeed very thin and they're unmyelinated. All right, so
actually ask like how it works when two types of things are connected. So for instance, like normally when I stretch my arms, it's not painful, but because I did rock climbing, now it's painful. Like why is it painful?
let me think about that.
Okay.
So if you have a stretch initially, it would not be painful. But now you have the same stretch.
And it's painful because of like the physical activity.
Okay, so I would guess it's probably not upregulation of B1 receptors. I would guess from my own experience more stuff like buildup of lactic acid or other things that could make it more sensitive to stimulating those. But let me look into it. That would be curious to find out. Certainly I'd love that when I try to get back to the gym and fail miserably. So we have a number of diseases of the peripheral nervous system. And these could include toxins, they could include metabolic diseases, genetic diseases, infectious agents, immune reactions, ischemia. What's ischemia? Low oxygen. Yeah, so lack of oxygen. Neoplastic would be, I heard it. Yeah, like a tumor or cancer. Traumatic injury, or again our favorite polyneuropathy. And so what we need to now consider is our diabetic patient. So we have a patient, and if you skip ahead, you'll know everything, but don't, because then, yeah, you can, that's fine. So he has tingling and numbness in the feet, and to a lesser extent in his hands. So why is he being tickled and you just don't see it? Peripheral neuropathy. Okay, we had it earlier, so someone knew earlier. So we have something going on. What's happening? Why would he feel tingling here? Yeah, good. So you have some damage to the fibers that are carrying those sensations. And what I'll tell you is that when neurons become hypoxic, which is often a response of a diabetic tissue, is that it becomes hypoxic. The neurons, they don't just die, they'll start to fire action potential. So they start to misfire, they start to misfunction, and that would account for the tingling there. So you have hypoxia, and the hypoxia is going to cause the tingling. And what's tingling is that what type of fibers would cause something like that? What is it? So some sort of sensory. So if you're on here, what could cause tingling? Not consciously perceived. So fine touch, vibration, and maybe some of our free nerve endings. But yeah, right in here. So fine touch, sort of vibration. And you can see that these are sort of in between. So these are sort of medium diameter fibers. Now we have an ulceration, and I mentioned to you that ulceration is often because you're just not feeling pain and you're walking on your foot funny, for example, and you're damaging, you're exacerbating whatever damage there is there, and then that leads to that ulceration. So there isn't pain associated with this. So let's go back, and what do you think? So we don't have pain here. So we probably lost these. So these, you had hypoxia, you lost them. There might have been a little pain associated with it, but not enough, not like the tingling where you have this sensation of pain. So you lose these, so you don't notice that you're walking funny, you don't notice you have a little cut, it develops into ulceration, you keep walking on it, and you have that ulcer, so you detect that. So you know you're here. You've started to stimulate some of these fibers, so not with their proper modality, you're stimulating them through hypoxia, and they're starting to fire. So you know they're still there because they're firing. Sure.
So when you do a monofilament test for diabetics, are you looking for two? Because they do it in early diabetics and late, so which?
So just describe the test.
It's literally just like maybe a fishing wire thing, and they just like run it along the foot and see which part of the foot they can feel and what they can't.
To poke it a little bit. So I'm not sure. I would have to look that up. So there, I would guess you're just looking to discriminate in here. You're looking to figure out whether you still have functional receptors here. So that would detect pain. What we have on our example is tests for these fibers, which would be next step. Sure, Dan. So the hypoxia comes from misregulation of glucose. So misregulation of glucose is going to induce hypoxia, so it's going to cause the blood vessels to thicken. The system I know the best is the retina, So in the retina, your vessels thicken, and then you actually don't get good diffusion of oxygen, and you stimulate growth of new vessels, and then those leak. And that's what causes the blindness there. But in general, the continuous state of high blood glucose will cause dysregulation of the vasculature, so it's not as good at transmitting oxygen to the tissues.
You're saying that in the patient, the A beta group is being stimulated by the hypoxia and not by its normal stimulus?
Yes, in this case, they could be stimulated by the normal
Okay.
stimulus. So if you were testing to see if they were still sensitive, then you would still feel that. So that would be the appropriate
The tingling
stimulation.
is coming from
The tingling is going to be from the hypoxia. So the hypoxia is going to damage the cells a little bit, and because they're damaged, they're going to fire. So it's a little bit like way back in the old days when you squeeze the axon to damage it and trigger a response. So a little bit of damage will do that. So we have our tingling, we have ulceration, but no loss of motor function, and your deep tendon reflex is normal. And so what can you tell me about those two bits of information? 1A and 1B are still there, so they're still working. And now put this together, and what can you tell me about hypoxia and neurons? Yeah, so the smallest neurons, the smallest diameter axons, are going to be the most sensitive. And so in this case, we've lost our sensation of pain. This has led to ulceration. We have some deficiency in fine touch, so we have some tingling because we're stimulating those, but our larger diameter axons are still intact. So this is caught sort of in a middle point where you have had some but not complete damage. And then that's just explained out here for you for when you review. So just the order that those would be damaged in. And here's our wonderful, for Caitlin, our wonderful unused slide this year, which is just the proportionality of the stimulus and the receptor potential. So it often is, it does correlate, but in some cases it will correlate and then plateau out. All right, that's it. Who's going to go water slide down the fraternities? I raised my hand. No, I won't. They always yell at me and say, do you know anything about private property? Because you walk up the hill. And I say, you mean the university property that is being maintained by the university that your fraternity's on? I always hope they'd be one of my students too. They haven't been, so. That's it. On Tuesday, we're going to talk about G protein coupled receptors.